Hello and welcome to our third video on Chapter 42 on the immune system. In this section, we are going to talk about antibodies. And our learning objectives are to explain cross-reactivity, describe the structure and function of antibodies, and discuss antibody production. So first, what is an antibody? We have been also calling these immunoglobulins, but they are proteins that are produced by our plasma cells after the B cells have been stimulated by that antigen. And they'll function within that humoral or antibody-mediated immunity, which we know is a subdivision of adaptive immunity. We'll find these antibodies within our blood, in our gastric and mucus secretions, and even within breast milk. They'll bind to pathogens to essentially mark them for destruction. It's kind of when they go through a forest and mark trees with an X so that they know which ones to get rid of, chop down. Hopefully that isn't happening anywhere where we have natural forest preserves, but you get the idea of where I'm trying to go with this. And so the phagocytes are the ones that are going to see those red X's and gobble them up. And so this plays into our passive immunity. This is when a person who has produced a successful immune response to a particular disease agent, so bacterial cell or virus, and they can donate blood to a non-immune recipient so that they can get temporary immunity through antibodies in the donor's blood serum. And this can also take place naturally through breastfeeding. And soon we're going to talk about a specific antibody known as IgA that is found within our secretions that is found in breast milk. And so that really helps to protect the infant from pathogens. So let's jump into the antibody structure. Our antibodies have this Y shape to them. Yes, this looks similar to our B cell receptor, right? But they are off on their own in the blood. And this is, they can also be within other secretions, by the way. They are soluble proteins, and we see 4 polypeptide chains here. We have 2 identical heavy chains. So this is our heavy chain, and we have 2 identical light chains over here. Now they will be flexible because they have a hinge region of those 2 heavy chains, and they're held together in the center with a disulfide bond. And this forms our antibody monomer. Remember, mono means 1, so this is 1 antibody here. And there are 2 important functional areas similar to our B cell receptor. We've got that constant region and the variable regions over here. So going into detail here, our variable regions are going to be the areas where we call them the arms that will bind the antigen. And most of them are going to have 2 sites. And we bind the antigens through weak intermolecular forces. This could be through hydrogen bonds, ionic bonds, and hydrophobic interactions. Whereas our constant region down over here is going to determine the biological function of the antigen. They'll have the same constant region depending on the class of the antibody. So we have 5 major classes. They are IgG, IgM, IgA, IgD, and IgG. Let's talk about each of those classes a little bit more. So we'll start off with IgG. This makes up the majority of the antibodies in our blood, making up about 75 to 80% of them. And it is known as our serum antibody because of this. We'll find it in other fluids as well, like our lymph and CSF, cerebrospinal fluid. And it can participate in all types of antibody actions. We'll talk a bit more about what those actions are in just a few slides. And it can also cross the placenta. It's the only immunoglobulin that is able to do this. And in the case of hemolytic disease of a newborn, unfortunately, it's because of this antibody that's able to cross that placenta. But luckily, we have really good screening now in order to prevent that from happening. The crossing of the placenta is great because we are able to give some immunity to the soon-to-be born infant. And then we have IgM. This is found mostly in our blood as well, and it's normally set up as a pentamer structure. So you can see that we have about 5 of these antibodies attached at the base over here, and it's the most effective at agglutination and binding complement proteins. So agglutination, remember we talked a little bit about this when we talked about blood typing, and if you give the wrong blood type, essentially these antibodies will clump together. They'll take the antigens of the red blood cells and clump them together and glue them, right? There's glue in there. And then the complement proteins, that is going to help mark the area for destruction as well. And we get into that a little bit later. And so they're going to be responsible for the rejection of mismatched transfusions. Next up is IgA, and this is found in areas exposed to the environment. So think a lot about those mucosal membrane tracts that we discussed. So we find it within the mucus, saliva, our tears, and breast milk. And like I said, our GI and respiratory tracts to protect them. And although it may be found as a monomer like this, it can be as a dimer here where it's composed of these 2 antibody molecules. It really helps to prevent the pathogens from adhering to and penetrating the epithelium. So we really want those secretions to be covered with things like the mucus or our oral cavity with saliva so that pathogens stay out of the lining there. And it's especially good at agglutination. Then we have IgD. Now this functions as an antigen-specific B lymphocyte receptor, essentially like a BCR, and identifies when our immature B cells are ready for activation. And IgE, this is usually found in response to parasites and in allergic reactions. Otherwise, we really don't see this synthesized by the body, and it's going to cause release of products from the basophils and the mast cells. So as you can imagine, this is really related to that allergic reaction of them releasing things like histamines. And it also attracts eosinophils, especially when we have a parasitic infection taking place. Now, there's a possibility of class switching when a plasma cell changes the type of antibody that it produces. This does require contact between that helper T cell and the plasma cell, and the T cell has to release a particular kind of cytokine in order to specify which antibody class needs to be formed. All right, and some of the antibody functions or actions here. So antibodies can help neutralize. You could see that taking place over here where we have a virus and the antibodies have attached to the protein spikes over here, whereas over here we have the diphtheria toxin and antibodies have attached there. By the antibodies attaching to those antigen determinants, it really is stopping it from being able to establish infection because it will need these structures to adhere to our tissues. Then we have the function of opsonization. This is when the FC region of our antibody classes makes it more likely as a target that the cell will see it. By cell, I mean an immune cell being able to see that pathogen so that a phagocyte can come over and gobble it up. This truly is like marking it with the red X so it can come on over and phagocytize. And then we have our complement activation. Now this is when our FC region of our IgG and IgM can bind to the complement for activation. And it goes through this process of classical complement activation. I didn't go into deep detail with this, but essentially what happens is they create something called a MAC complex, membrane attack complex. And those complement proteins kind of create pores within the pathogen, and water will move in and cause the cell to lyse. Now, your book talks about these 3 main functions, but we also know that antibodies are going to help with agglutination, that clumping. They can also work in something called precipitation, where an antibody is going to cross-link circulating antigens and will form a complex so that it becomes insoluble and will precipitate out of our body fluids. At that point, a phagocyte can come around and gobble it up in order to eliminate it. And lastly, antibodies can also help activate our NK cells through something called ADCC, and this is called antibody-dependent cell-mediated cytotoxicity. Essentially, the IgG antibodies will trigger the NK cells to release cytotoxins, and they will be destroyed through that process. Now let's talk about a few of these terms here. When we say affinity, we are talking about the attraction between the antibody and the antigen. This is going to depend on the molecular complementarity of these two structures. And if we have a high affinity, that means we have stronger bonding and stability here. Next is avidity, and this is the binding of that antibody classes that are secreted as joined multivalent structures. So this works best with IgM, which is pictured over here, and you can see that we've got the multiple antibodies attaching, and IgA because it is a dimer. So it does depend on the number of identical binding sites and the antigen that is detected. Lastly, we have cross-reactivity, which we have pictured over here, which is when the antibody is going to bind not to the specific antigen that it was made for. And I think this picture did get cut off a little bit, but notice how the epitopes here are a little bit different, right? And the antibody is still able to attach to both of them. So in this way, we are able to develop immunity to several related pathogens, and different macromolecules can exhibit the same molecular identities. For instance, we may have antibody reactivity against similar surface structures such as gram-negative bacteria because they are going to have similar structures along their plasma membrane. And then we've got our antibodies of the mucosal immune system. This is going to include IgA and IgM, and it helps to activate the B cells differentiate into mucosal plasma cells that'll synthesize and secrete either dimeric IgA and to a lesser extent that pentameric IgM. And this helps to prevent infection of that mucosa by binding and neutralizing pathogens. And the last thing to mention is antibodies in detection and measurement. We have this radioimmunoassay, or RIA, in which we are going to rely on that antigen-antibody interaction to be able to detect the presence and or the concentration of certain antigens. So we can see this taking place over here. This was developed by Rosalind Sussman Yellow and Solomon Barrison in the 1950s, and Yellow received a Nobel Prize, which made her the second woman that was awarded the prize for medicine. So we now use a system in narcotics detection, blood bank screenings, early cancer screenings, hormone measurement, and allergy diagnosis. Essentially what takes place here is we have a radioactive antigen that is going to compete with the non-radioactive antigen for a fixed number of antibody or receptor binding sites. And so we could take our sample, for instance, a patient's blood, and the antibodies will cease binding to the tagged antigen and instead bind to the untagged one, which is called displacement. After a few steps, then we are going to look at the amount of unbound tagged antigens so that we can see how many were displaced, which will indicate for us the presence and concentration of the antigen within the blood.